Hello friends, this is Lieutenant Dr. Yasin from Abeda Namdar Senior College. Today we are going to discuss about how macromolecules, that is having the cell substance, is going to divide into different biomolecules. There are the four different and very important structure of the macromolecules, that is the nucleic acid, the proteins, lipids and the carbohydrates. Now today we are going to discuss about see the how it's going to be defined. The water is definition of water. It makes 100% of the cells volume. Nucleic acid, long chains of nucleosides. Proteins are the long chains of amino acid, and the lipids are the large macromolecules that does not dissolve in water. How example is going to be fats and oil, enzymes and air in the proteins, and RNA and DNA in nucleic acid, and importance of the cell as well. Now we are going to do a very important very important topic, that is a nucleic acid. The central dogma of the molecular, you can see the replication process, the DNA, which can be go for the protein, and the translation RNA, and its transcription is going to be a DNA. This is the the central dogma of the molecular structure. Now nucleic acids and its heredity, how it's going to be processed. The transfer of genetic information is a replication that identical copies of the DNA are made. The transcription, the genetic message are read and carried out of the cell nucleus to the ribosomes where protein synthesis occurs. Now translation is the last process that genetic messages are decoded to make the proteins as well. Now you can see the definition of the nucleic acid are polymers of nucleotides, and its carbon ring structure containing nitrogen linked with the 5-carbon sugar and ribose. 5-carbon sugar is either ribosomes or deoxyribose, which makes the nucleotide either ribonucleotide or deoxyribonucleotide. In eukaryotic cell, as we have discussed in the last lecture, its nucleic acid are either deoxyribose, that is the DNA, or a riboxylic nucleic acid, that is the RNA. And RNA contains messenger RNA, transfer RNA, and ribosome RNA, as well as the you can see mRNA, dRNA, and tRNA. Now you can see the D nucleic acid function, how it can act. Genetic material sequences of the nucleotides, it encodes the different amino acids. RNA, which involves in the transcription and the translation of the genetic materials, that the genetic material is of some viruses. Now you can see the structure of the nucleotides. Despite of the complexity and the diversity of a life, with the structure of the DNA is going to be dependent only on the 4 different nucleotides. That is, diversity is dependent upon the nucleotide sequences. All nucleotides are 2-ring structure composed of 5-carbon sugar, base, and phosphate group. 5-carbon sugar is the beta D dibose RNA, beta D deoxyribose RNA, DNA, and base is what? Purine and pyrimidine. Purine and pyrimidine. Phosphate group, which can nucleotide without a phosphate group, is a nucleoside. It is very important. Here we can see the table is given in the nucleoside and nucleic acid nomenclature. You can see the purine and the pyridine and guanine and pyrimidine. These are the different. You can see the names of the nucleosides and nucleotides as well. Adenine, adenine A, adenine, guanine, and the nucleosides and nucleotide without phosphate group is as well. You can see the base, purine, pyrimidine, and plus ribose, deoxyribose, which is going to be glycosyl linkages, nucleosides and phosphate, which is going phosphater linkages, and nucleotides phosphodiester linkage. Now you can see the functions of the nucleotides and nucleic acid. Now you can see the nucleotide function. It can be energy for a metabolism, be it ATP, enzyme cofactor, that is a NAD plus, and signal transduction, that is a CAMP. Nucleic acid functions, that is the storage of the genetic information, that is a very important and very important. DNA transmission of the genetic information, that is mRNA. Processing of the genetic information, that is the ribosomes, and protein synthesis, that is the tRNA and rRNA. The base pairing in the DNA, the Watson and Crick model, that shows the DNA that in 1953, the Watson and Crick noted that DNA consists of 2 polynucleotide strands running on the process in opposite directions and coiled around each other, and the double helix structure. The strands are held together by hydrogen bonds between the specific pairs of bases, that is the adenine and thymine. The thymine are for strong hydrogen bonds to each other, but not to C or G, that is the cytosine, and the G is the for that form. Guanine and the cytosine are the form strong bonds to each other, but not A or T. That there's a strange difference between the strands are there. So strands of DNA are complementary because H bond, hydrogen bonding is in the action. And whenever the G occurs, that is the union occurs in one strand, that the cytine also occurs in the opposite in the either strand. When an occurs, adenine is occurs in one strand and the thymine is occurs in the other. These are the, you see the 3-5 phosphate ratio. We can see show you the base and the structure is given to the action. The primary structure of the nucleic acid is nucleotide sequences. The nucleotide is a nucleic acid which joined by the phosphodiester bond, and 3 OH group of the sugar in one nucleotide forms an ester bond to the phosphate group as well on the 5 carbon of the sugar in the next nucleotide. You can see the formation of the ester bond to the first 5 phosphate, and the phosphodiester bond is given to the action. You can see you can go with the diagram and you can easily predict the sugar and the bases of molecule, how it's going to be happen with the 3 hydroxyl groups are there. Now you can see the generalized structure of DNA. It's openly, I have given you the structure, very, very nice. This 5 end, you can see the 5 end is phosphate, sugar base, phosphate, and sugar base. That is the 3 end. And you can see the 5th position of the carbon and the third position of the carbon as well. Now you can reading primarily structure of how you can read the nucleic acid polymer has a free 5 phosphate group that at one end you add the free of 3 OH group at the other end. So you can see the one end, the one group is there and the another is the 3 OH group is there. And the phosphate group and OH group is combination into sequences that read into the form action. That is a 5 many using the latest of the bases. That is the example, right? 5ACETD. That is the, you can see into the action that phosphate and the thymine is into the last. Is always grouped into the 5 and 3rd carbon. You can see the properties of the DNA helix structure. The strands of DNA are anti-parallel, the strands of complementary, and there are the hydrogen bond forces, there are the base stacking interaction, and there are the 10 base pairs turned into the action of the. These are the untwisted. It looks like this, when the sides of the ladder pair. I told you before that's the phosphate and the sugar molecule is there. The steps into the ladder of CGTA. That is the, whenever the nitrogen is means containing the elements nitrogen, the adenine and the thymine. That's you can do. Apples are tasty. You can say adenine and thymine. A always pairs with the T in the DNA. C always pair with the G. That is the cookies are very good. You can say like this. C always pair with the G. These are the different combination. You can remember very well. Now second structure of the double DNA double helix. The DNA 3. There are the 2 strands of nucleotides that wind together and the double helix. The strands run in opposite direction. The base are arranged in the shape-like manners, with shape-like pairs. The base pairs are held together by hydrogen bonding, and the pairing of the bases are from the 2 strand in a very specific way. That is the complementary bases pairs. That A always apples are tasty, and the G and C. The 2 hydrogens are formed between A and T. 3 hydrogens are born from between G and C. Each pair consists of purine and pyrimidine, so they are the same width and the keeping into the 2 strands at equal distance from each another. You can sense versus the antisense DNA strand. The DNA double helix has 2 strands. Only one of them is transcript and the other transcript strand to the antisense strand, and the non-transcript strand in the sense that strands the mRNA is always complementary to the antisense strand. So these are the very easy way method of remembering the things. Now you can see the ribonucleic acid. This is the DNA. We finished with the DNA. Now we can go with the RNA. RNA is a much more abundant than DNA as well. There are several important differences between the RNA and DNA. The pentose sugar in RNA is ribose. In DNA, it's a deoxyribose. In RNA, uracils replace the base thymine. That is a U pairs with A, and RNA is single strand, while DNA is a double strand. And the RNA molecules are much smaller than the DNA molecules. And this are 3 main types of RNA. That is a ribosomal rRNA, messenger mRNA, and transfer. That's a tRNA. This is the types of RNA. I will show you the abbreviation also. The percentage of total 75 in the RNA, ribosomal MS, messenger में 5 to 10 and transfer in the 10 to 15. What's the function of the cell? Major component of the ribosomes is the RNA. Carries information from protein synthesis from the DNA is in the nucleus to the ribosome is a function of the mRNA, and ultimately the tRNA brings amino acid to the ribosomes for protein synthesis. These are the sequences copied from the genetic DNA. It travels to ribosomes, smaller granular particles to the cytoplasm and cell where protein synthesis is taking place. RNA is a complex of a proteins and RNA. The synthesis of proteins from amino acids and ATP occurs in the ribosomes. The rRNA provides you the both structure and the catalysis. The tRNA transport the amino acid into ribosomes where they are joined together to make proteins. There are the specific tRNA for each amino acids, and the recognition of the RNA tRNA is the anti-codon communicates which amino acid is attached. The transfer of RNA is to translate the genetic code from the messenger RNA. It brings the specific amino acid to the ribosome for a protein synthesis. And one end attached to the amino acid and the other binds to the mRNA, which can give you the three bases of the complementary sequences. This is the very, very important these are fundamental properties of the RNA. You can see the ribosomes are the sites of protein synthesis. They consist of hydrogen, ribosomal DNA 65%, and the proteins are the 35%. They have two subunits. A large one is one small one, and the messenger RNA carries the genetic code to the ribosomes. They are strands of the RNA that are complementary to the DNA of the gene of the protein to be synthesized. Here we can see the action of this small subunit, large subunit, and ribosomes as well. So here we can finish with the nucleic acid, and if you have any question, any doubt, you can ask in that question box. Thank you so much. Be patient. Thank you. God bless you.